The Golgi complex, also called lipochondria or idiosome or Dalton complex or Golgi body or Golgi apparatus, was first observed by St. George in 1867. Holmgren in 1900 described a system of clear canals and named it as tropospongium. Pironcito in 1910 named it as dictyosome and their division as dictyokinesis. Baker in 1951 described it as lipochondria and later in 1959 as Dalton complex. However, the term Golgi complex has been widely accepted. In 1898, Camillo Golgi, an Italian neurologist, communicated his finding of a novel intracellular structure which he had detected in nerve cells of cats and barn owls by means of a variant of black reaction he had developed earlier for the staining of the nervous tissue. On the basis of the net-like appearance and intracellular location of the structure, Golgi defined it as apparato reticular interno, means internal reticular apparatus. The reality of the organelle was debated for 50 years, since some investigators believed that structure, which was soon designated as Golgi apparatus, represented an artifact of metallic impregnation. The controversy was finally solved in mid-1950s by studies based on electron microscopy. The Golgi possesses 4 types of components, that is cisternae, tubules, vesicles and vacuoles. Now, first of all, let's discuss briefly about cisternae. Cisternae are membrane-bound, curved, flattened sacs which occur in stakes of 5 to 8. The curvature gives a polarity to cisternae. There is a convex, formative phase, and concave, that is maturation phase. The concave phase is towards plasma lemma. A cisternae encloses a space or lumen of 5 to 10 nanometers width. Now, tubules, they are short, branched, interconnected, hollow, filamentous structures which develop on the sides and maturing face of cisternae. A tubule is 30 to 50 nanometers in diameter. Now, vesicles, they are small sacs which develop from the tubules. They contain secretory products and are therefore called secretory vesicles. These vesicles are of 2 types, as smooth and coated vesicles. These vesicles bud off from the tubules within the network. On being pinched off, they pass on to either cell membrane or throwing out their products through exocytosis or to an intercellular region for a specific activity. Now, vacuoles. Vacuoles are large, round vesicles which are produced by the concave maturing face of cisternae. Animal cells generally contain between 10 and 20 Golgi stakes per cell, which are linked into a single complex by a tubular connections between cisternae. This complex is usually located close to the cell nucleus. A mammalian cell typically contains 40 to 100 stakes. Between 4 and 8 cisternae are usually present in a stake. However, in some protists, as many as 60 have been observed. Each cisterna comprises a flattened membrane disc and carries Golgi enzymes to help or to modify cargo proteins that travel through them. They are found in both plant and animal cells. The cisternae stake has 4 functional regions: the cis-Golgi network, medial Golgi, endo-Golgi, and trans-Golgi network. Vesicles from the endoplasmic reticulum via the vesicular tubular clusters fuse with the network and subsequently progress through the stake to the trans-Golgi network, where they are packaged and sent to the required destination. Each region contains different enzymes which selectively modify the contents depending on where they reside. The cisternae also carry structural proteins important for their maintenance as flattened membranes which stake upon each other. Due to its relatively large size, the Golgi apparatus was one of the first organelles ever observed. The Golgi apparatus is often considered the distribution and shipping department for the cell's chemical products. It modifies proteins and lipids or fats that have been built in the endoplasmic reticulum and prepares them for export outside of the cell or for transport to other locations in the cell. Proteins and lipids built in the smooth and rough endoplasmic reticulum bud off in tiny bubble-like vesicles and move through the cytoplasm until they reach the Golgi complex. The vesicles fuse with the Golgi membranes and release their internally stored molecules into the organelle. Once inside, the compounds are further processed by the Golgi apparatus, which adds molecules or chops tiny pieces of the ends. When completed, the product is extruded from the Golgi apparatus in a vesicle and directed to its final destination inside or outside the cell. The exported products or secretions of proteins or glycoproteins that are part of the cell's function in the organism. Other products are returned to the endoplasmic reticulum or may undergo maturation to become lysosomes. The modifications to molecules that take place in the Golgi apparatus occur in an orderly fashion. Each Golgi stake has 2 distinct ends or faces. The cis face of a Golgi stake is the end of the organelle where substances enter from the endoplasmic reticulum for processing, while the trans phase is where they exit in the form of smaller detached vesicles. Consequently, the cis phase is found near endoplasmic reticulum, from where most of the material it receives comes, and the transphase is positioned near the plasma membrane of the cell, to where many of the substances it modifies or shipped. The chemical makeup of each phase is different, and the enzymes contained in the lumens of the cisternae between the phases are distinct to proteins, carbohydrates, phospholipids, and other molecules formed in the endoplasmic reticulum are transported to the Golgi apparatus to be biochemically modified during their transition from the cis to the transpoles of the complex. Enzymes present in the Golgi lumen modify the carbohydrate or sugar portion of the glycoproteins by adding or subtracting individual sugar monomers. In addition, the Golgi apparatus manufactures a variety of macromolecules on its own, including a variety of polysaccharides. The Golgi complex in plant cells produces pectins and other polysaccharides specifically needed for plant structure and metabolism. The products exported by the Golgi apparatus through the transphase eventually fuse with the plasma membrane of the cell. Among the most important duties of the Golgi apparatus is to sort the wide variety of macromolecules produced by the cell and target them for distribution to their proper location. Specialized molecular identification labels or tags, such as phosphate groups, are added by the Golgi enzymes to aid in this sorting effort. Cells synthesize a large number of different macromolecules. The Golgi apparatus is integral in modifying, sorting, and packaging these macromolecules for cell secretion or use within the cell. It primarily modifies proteins delivered from the rough endoplasmic reticulum, but is also involved in the transport of lipids around the cell and the creation of lysosomes. In this respect, it can be thought of as similar to a post office. It packages, labels items, which it then sends to different parts of the cell. Enzymes within the cisternae are able to modify substances by the addition of carbohydrates, glycosylation, and phosphates, phosphorylation. In order to do so, the Golgi imports substances such as nucleotide sugars from the cytosol. These modifications may also form a signal sequence which determines their final destination. For example, the Golgi apparatus adds a mannose 6-phosphate label to proteins destined for lysosomes. The Golgi plays an important role in the synthesis of proteoglycans, which are molecules present in the extracellular matrix of the animals. It is also a major site of carbohydrate synthesis. This includes the productions of glycosaminoglycans or GAGs long unbranched polysaccharides, which the Golgi then attaches to a protein synthesized in endoplasmic reticulum to form proteoglycans. Enzymes in the Golgi polymerize several of these GAGs via xylose link into the core protein. Another task of Golgi involves the sulfation of certain molecules passing through its lumen via sulfotransferases that gain their sulfur molecule from a donor called PAPs. This process occurs on the GAGs of proteoglycans as well as on the core protein. The level of sulfation is very important to the proteoglycans, signaling abilities as well as giving the proteoglycan its overall negative charge. The phosphorylation of molecules requires that ATP is imported into the lumen of the Golgi and then utilized by resident kinases such as casein kinase 1 and casein kinase 2. One molecule that is phosphorylated in the Golgi is apolipoprotein, which forms a molecule known as VLDL, that is a constituent of blood serum. It is thought that phosphorylation of these molecules is important to help aid in their sorting for secretion into the blood serum. The Golgi has a putative role in apoptosis with several BCL2 family members localized there as well as to the mitochondria. A newly characterized protein GAAP, that is Golgi anti-apoptotic protein, almost exclusively resides in the Golgi and protects cells from apoptosis. The vesicles that leave the rough endoplasmic reticulum are transported to the cis face of the Golgi apparatus, where they fuse with the Golgi membrane and empty their contents into the lumen. Once inside, they are modified, sorted and shipped towards their final destination. As such, the Golgi apparatus tends to be more prominent and numerous in cells, synthesizing and secreting many substances. Plasma B cells, the antibody secreting cells of the immune system, have prominent Golgi complex. Those proteins destined for the areas of the cell other than either the endoplasmic reticulum or Golgi apparatus are moved towards the transphase to a complex network of membranes and associated vesicles known as trans-Golgi network or TGN. This area of the Golgi is the point at which proteins are sorted and shipped to their intended destinations by their placement into one of at least 3 different types of vesicles, depending upon the molecular marker they carry. The transport mechanism which proteins use to progress through the Golgi apparatus is not yet clear. However, a number of hypotheses currently exists. Until recently, the vesicular transport mechanism was favored, but now more evidence is coming to light to support cisternal maturation. The 2 proposed models may actually work in conjunction with each other rather than being mutually exclusive. This is sometimes referred to as the combined model. Now, cisternal maturation model. The cisternae of the Golgi apparatus move by being built at the cis phase and destroyed at the trans phase. Vesicles from the endoplasmic reticulum fuse with each other to form a cisterna at the cis phase. Subsequently, this cisterna would appear to move through the Golgi stake when a new cisterna is formed at the cis phase. This model is supported by the fact that structures larger than the transport vesicles, such as collagen rods, were observed microscopically to progress through the Golgi apparatus. This was initially a popular hypothesis but lost favor in 1980s. Recently, it has made a comeback as laboratories at the University of Chicago and University of Tokyo have been able to use new technology to directly observe Golgi compartments maturing. Additional evidence comes from the fact that copy vesicles move in a retrograde direction, transporting endoplasmic reticulum proteins back to where they belong by recognizing a signal peptide. Now, vesicular transport model. Vesicular transport views the Golgi as a very stable organelle divided into compartments in the cis to trans direction. Membrane bound carriers transport material between the endoplasmic reticulum and the different compartments of the Golgi. Experimental evidence includes the abundance of small vesicles, known technically as shuttle vesicles, in proximity to Golgi apparatus. To direct the vesicles, actin filaments connect packaging proteins to the membrane to ensure that they fuse with the correct compartment. The Golgi apparatus will break up and disappear following the onset of mitosis or cellular division. During the telophase of mitosis, the Golgi apparatus reappears.